Hey everyone, welcome to the lab design in AP Biology. I want to welcome you to an exciting school year, and I think that this sheet historically has been pivotal in helping students understand how to do the lab design process in AP Biology, as well as help them with writing labs, posters, FRQs, answering multiple choice questions. We try to break it down so that it is clear and easy to understand. In this video, I'm going to be going over the first two topics, the question and the hypothesis. So let's get started. Starting with section 1, the scientific design question. It's going to give you an idea of what is being tested, and it needs to be set up in such a way that the independent and dependent variables are clearly identified. So to make life simple, we provide this template example for you. It doesn't have to be exactly like this. However, when you're writing scientific questions, if you don't write them in the proper format, you won't get credit for them. So we find that this way is the easiest way to help you with setting up your lab design and the following sections much later. The template goes like this. How does blank change in response to blank? Now what we're going to fill in here is, how does the dependent variable change in response to the independent variable? Now your independent variable is the part that you are designing in your lab. You have the power to control what you're going to be testing with this independent variable. And the dependent variable is going to be the results, what you want to see, does this change? So let's take a look at an example. So the question is, how does the number of eggs for turtles change in response to temperature? Based on this format, we can clearly see what the independent variable is going to be. So we're going to be changing the temperature that the turtles are living in. And in response, we want to see, does the number of eggs change for each turtle at the different temperatures? Now, the question doesn't have to be in this format. The part that you need to understand is that the question focuses on your independent and dependent variable. And the way that the question worded may be different. So, for example, this question could have been phrased, how does temperature affect the number of eggs laid by turtles? But you can still clearly see our independent variable of temperature and our dependent temperature, which is the number of eggs. So either format is going to work. You've just got to make sure you clearly have a dependent and independent variable identified. Let's practice this some. In the next part of this lab design process, we're going to be talking about the hypothesis. Now, normally your hypothesis takes the independent variable and dependent variable and predicts what the effect or relationship would be. It's going to focus on the effect between the independent and dependent variable. So, for example, you could say this increases that, or something is going to decrease in response to something else. You can either also say promote or inhibit, or you can even say, such as the state of a null hypothesis, that there's going to be no difference between the specified populations. In other words, that this is not going to be any different from that, or A is going to have the same results as B. So a null hypothesis is a very specific type of hypothesis, and it's used in statistical testing, and it assumes that there's no difference between specified populations. So let's look back at our previous example with temperature and the number of eggs laid by a turtle. So if we were to make a null hypothesis, the way that we do that is we indicate by writing H subscript O. This stands for the null hypothesis. So our null hypothesis is going to be that temperature does not affect the number of eggs laid by the turtle. And then opposite to this is the alternative hypothesis. And so this would state that the temperature does affect the number of eggs laid by turtles. And so these two are complete opposites. They can't both be true at the same time. One of these is going to get rejected. And so our data from our experiment is going to support either the null or the alternative hypothesis, not both. Thank you so much for watching. This concludes part 1 for the lab design topics in AP Bio. Bye.